So you're stuck with me, just a grad student, so I'll try my best to describe the experiments as best I can. It's very cool stuff, so hopefully I'll do it justice. So the main goal of our lab is to actually find signatures within neurodegeneration. Mainly we work on Huntington's disease, but I'll go over in the talk a few other diseases as well too. And so our lab uses a bunch of different types of techniques to actually find these signatures. But lately, and what I'll be going over in this talk, is defining transcriptomic signatures within neurodegeneration. So first, a little bit of background about Huntington's disease, because that will be the bulk of the talk. It was first characterized by George Huntington in 1872 in a publication titled On Korea, because of the dance-like movements that the individuals that have when they have Huntington's disease, which we'll see in the next slide. It's a progressive neurodegenerative disease. As I stated, there are uncontrolled movements, as well as some other symptoms are psychiatric, as well as cognitive deficits. What's unique about Huntington's disease is that, unlike a lot of the other neurodegenerative diseases, we actually know the gene that causes it. So it's one gene, which is entitled interesting transcript 15, or the Huntington gene. And this encodes for the Huntington protein. It's a very huge protein, and we actually don't know the normal function of it as of yet, although many people are working on that right now. They're very close to figuring that out. But within this gene, there is a trinucleotide repeat, a CAG repeat, in the first exon. And this encodes for a poly-T track. And normal individuals have around 6 to 34 repeats. But any individual that has over 40, if you live long enough, you will get the disease. There are individuals that have very high repeat levels, and this correlates with the onset of the disease. There are juvenile onset cases, excuse me, that are below the age of 20. And one really interesting case where an individual had 180 repeats, and they ended up getting the disease at a very, very young age. One of the characteristics of the disease is loss of DARP32 positive medium spike neurons in the striatum, which is shown here. You can see that there's massive loss in the striatum in this region. And there's also atrophy within the cortex too, but the striatum is mainly the affected area. This movie should be actually playing right now. I'm not sure why it's not. But as you can see, this is an HD patient, and you can see they have the dance-like Korean movements, as well as a characteristic wasting of the body. And so it's a very, very devastating disease, unfortunately, and affects a lot of people in the U.S. as well too. This is one family that we work with closely. We have a lot of interaction with them. They're advocates for other HD patients. The father had HD, the mother did not, and they had 3 children, and unfortunately all 3 children ended up getting the disease. And so it's very, very devastating and motivates us as scientists to actually try and find a cure, which is the reason why we're modeling disease and trying to find cures and therapies right now. So our lab is part of an international consortium, the HD IPSL consortium, where labs across the US as well as internationally are all working to utilize iPSCs to model the disease. And so this collaboration has been going on for a few years. Here at UCI, we've also collaborated with Peter Donovan and Leslie Locke to actually generate the iPSCs. And the accumulation of all that work led to a publication in 2012 that actually showed after the neural stem cell stage there were deficits. So the HD patients, they were all juvenile repeat length cells, had 60 plus repeats, actually showed a phenotype, which is very, very interesting. Since then, we moved on to more adult populations, or adult neuronal populations, with striatal-like characteristics. And so the approach right now in the lab is basically outlined here in this flowchart, is to generate medium spiny neurons, or striatal-like cells, as well as astrocytes and even brain endothelial cells. And taking all these cells and doing various functional assays, as well as multi-omic analysis on these cells. So mainly here at UCI in our lab, we do RNA-seq to analyze the transcriptome. We are generating these primate signatures for each of these cell types, and then using an iterative process where we're building signatures using multicellular analysis as well as functional experiments and in vivo experiments to guide us for signature verification and then back to signature building. And so that's the overall flow and the approach that we're taking. Just very briefly, the project that I mainly work on in the lab is with the blood-brain barrier. So utilizing those iPS cells to make brain endothelial cells and analyzing are there any alterations. I've been working on this project for a few years now. Interestingly, a paper just got published this month showing that there are deficits of the blood-brain barrier in HD. Like I said before, I just want to spend a moment on this. If you have questions, I can go into further details after the talk. So going on with the main bulk of the presentation is the neuronal work. And so these are the patient samples that we've analyzed thus far. 3 of them having HD with very high repeats, and 3 of them as controls. They were all grown and differentiated in Clive Spence's lab at Cedars-Sinai, and then distributed to everyone in the consortia for various omics analyses. We did the RNA-seq. Future plans are to do knockdown with ASOs in these cells as well too. So for the RNA-seq, you can see that there's clear differential expression between the HD and control cells, the HD on the top and the control on the bottom. And then when you actually look at the differentially expressed genes and do some pathway and network analysis, very, very interesting things come up. 2 things. One, and probably the thing that we're most worried about, was do we see recapitulation of the disease? And we actually do. So a lot of the things, these are pathways up top, and here are some of the upstream regulators that are predicted to be activated or inhibited by our gene list. You can see things that make sense in HD already. For example, Huntington is predicted to be activated, as well as REST, which is a neuronal silencing gene, is predicted to be activated, and was already been shown in patients and animal models to be activated. And so the fact that we're recapitulating that in our iPS cells is really, really wonderful. In addition to that, you can see some of the very interesting pathways that are popping up, such as axonal guidance signaling, when signaling is affected, calcium signaling, glutamate receptor signaling, and GABA, which again is very interesting within HD because the medium spike neurons are mainly GABAergic neurons, and so that makes sense. In addition to that, novel things have popped up as well too, such as right here, this NeuroD1 gene, which you can see is very downregulated in our data set. And so we started to focus on that, and I'll go into that in a little bit further in the presentation. Here's just an example of the calcium signaling pathway. It's very highly impacted. There are a lot of genes that are impacted in this, and so we think that it plays a major role in disease pathogenesis. We also looked at functional categories, so these are kind of like go categories that pop up. And one interesting thing that we noticed was a lot of developmental categories popping up, and specifically nervous system function and development. As you can see, development of neurons, synaptic transition, even at the organismal level, physiological level, you see things impacting learning and memory and emotional behavior and things like that. So it's really, really interesting the things that are popping up. One of our collaborators is actually looking at striatal development in mice, and so development of the LGE into the GZ and MZ, which later on becomes these adult striatum. And so we can see when we take the genes that are differentially expressed in our human iPS cells and compare them with these cells, you can see that, interestingly, the HD cells cluster with the younger LGE and the GZ region, whereas the control are clustering with the MZ. And so that's really interesting, considering the fact that in the previous slide I showed you that there might be some type of developmental impact to these cells as well too. This basically shows some of the ChIP-seq work that's been done, complementing the RNA-seq work. You can see classically what ChIP-seq's known for is actually looking at the different types of marks. Here's an example of the H3K27 acetylation. And you can see in the control sample on top in blue, the higher mark level, its activating mark, correlates really well with the higher levels of RNA reads in the control on the bottom, and the HD is in red. But in addition to that, what our collaborator has also done at MIT in Earl Frankel's lab is looked at the way that the actual histone marks are sitting on the genes, sitting on the transcription start sites. They analyze different shapes. As you can see, this blue shape right here, which is termed class 1. There's 5 different classes that they categorize, and they predict how well does this define the differentially expressed genes that we see. And as you can see, they're very significant within class 1 for the K4 ME3 mark, as well as the K27 acetylation. And so this is really interesting, the fact that for some reason the genes that have this very specific shape right here and here in blue in the control cells end up being the genes that are dysregulated in HD, in our HD samples. And so somehow it primes those genes for dysregulation. It's something that we're investigating further. So back to NeuroD1, that gene that I highlighted in the upstream regulator list. You can see right here, one of the reasons why we're focusing on it and we're so excited about it is because it fits very nicely into this network as a hub. It shows genes that are very important already to Huntington's disease pathology, as well as novel things as well too, such as ASCL1 and genes that are related to striatal development that have been known already. As you can see, this little publication right here, NeuroD1 is essential for the survival of and maturation of adult neurons. On the bottom, it shows some qPCR data that shows that it's down in the HD samples compared to the control. This is the repeat length. And it's even down in the R62 mouse model of HD, which you can see here. So it seems to be recapitulating really well across everything. And so we focused on that gene, and we went through the literature and found that there's this one novel small molecule called ISX9 that actually can activate NeuroD1 through calcium signaling. And it's been shown in previous publications to actually activate neuronal cell fate. And so we utilized this molecule as a proof of principle to see, can we actually reverse the gene effect, the differential expression of NeuroD1, and the functional readouts that we also saw in the HD cells. And it looks like we can. Not only did NeuroD1 expression jump up with the treatment of ISX9, but in addition to that, a bunch of the functional readouts actually were reversed as well too, such as increased cell death was reversed after ISX9 treatment, as well as ATP deficits, and even glutamate excitotoxicity in these cells. And so it seems to be working really well, which is very exciting for us because it's very unknown whether or not once an individual gets symptoms, can you reverse those and can they be made healthy again. So in summary, for the HD work, the transcriptomic data and the ChIP-seq data basically correlate really well with different functional readouts, and they suggest that there's alterations to neuronal development and maybe even adult neurogenesis, which is something that hasn't been shown in HD within the striatum. We hypothesize that neurodevelopment and maturation is affected, but that effect can be tolerated for years through compensatory mechanisms, which is why you do not see any symptoms in individuals until they get older. Of course, that correlates with CH2 repeat length. But once they do get older, or some other type of factor that's involved, maybe environmental, the toxic impact of mutant Huntington cannot be compensated anymore, and that's when you end up getting neurodegeneration. This, coupled with impaired striatal neurogenesis, we think leads to actual disease itself, phenotypic disease. But as an outlook, we are very pleased, and we think it's promising that ISX9 data does provide proof of concept that you can treat terminally differentiated cells. And so maybe we can actually develop treatments and cures that can help these patients even when they are symptomatic. And again, like I said, future studies are going to look at knockdown of the mutant Huntington gene as well. So very briefly, I'm going to move from that work into a new project that kind of spawned from that. So a lot of the collaborators that were on or part of that consortia were asked to join this NIH-linked consortia, which is basically a library of integrated network-based cell signatures. Basically a bunch of groups across the US that are actually utilizing different omics techniques to actually generate cell signatures. And basically, we're looking to define what makes a specific cell type that cell type through transcriptomics and genomics and then different things like that. And when we perturb these cells, what happens to that signature? And so for our group, we're part of NeuroLinks. We're actually taking iPS cells, differentiating them into motor neurons first, defining what that signature is for those specific motor neurons. And then our perturbogen initially is going to be a disease state. So we're going to be looking at ALS and SMA patient samples. And so as you can see, a lot of the PIs that are on this group are similar to the ones that we had in the IPS consortium. We have a bunch of different iPS cell lines, but we're going to be starting with ALS and SMA, which are both motor neuron diseases. Here's some of the sources of tissue that we're getting the cells from. And this is the data generation flow. So these are the types of assays that we're going to be doing to actually define these signatures. And so you can see here that seeders, again, will be responsible for propagating cells and the differentiation itself. They'll distribute all the cells to the other labs. And each lab is responsible for a specific unique assay At UCI, we'll be doing the transcriptomics again. Cedars and Johns Hopkins will be responsible for perturbagens. At the Gladstone Institute, they're doing very unique single-cell imaging on neurons. And then also at Cedars in Jenny's Van Eyck's group, they'll be doing proteomics, as well as we added on lipidomics and metabolomics to this study as well too. And LIT will be doing epigenomics, utilizing the CAT seeds for chromatin states. And so those are the initial cell signatures that we're going to develop through those assays, and then we're going to take all the cell signatures and then hopefully integrate them all together utilizing different network and pathway-based analysis as well as machine learning techniques. So thus far, basically we haven't generated too much data yet. It's been a lot of getting things going and knowing what we're going to have to report and things like that. But some of the data that we did generate, I'm displaying here. You can see these are PCA analysis of disease and control iPS cells for the ALS samples, SMA, and control. This is our RNA-seq data that we generated thus far. And you can see clearly what's really interesting is that even at the iPS cell level, there might be some differences, which is something that we did not see in the HD cells. And so along principal component 1, which is turned away from me right now, but along principal component 1 right here, you can see that the majority of the variability along principal component 1 seems to be with the patient samples themselves. And so you don't see a large amount of variance between any individual group, but you see the samples for each individual patient scattering across the entire principal component, which is what you would expect at the iPS cell level, maybe, especially considering our HD data. And then, but in addition to that, the second principal component 2, which is up here, the y-axis, you can see that there actually is separation between the ALS group and the SMA group and the ALS control, which seem to cluster together on the top. You can still kind of separate those groups away from each other, but not as clearly as the SMA group, which is down at the bottom. And so what's even more interesting is the fact that the SMA disease actually affects is childhood disease. So I don't know if that's going to have any role in actually seeing symptoms at the iPSL stage, but it possibly can. So it's something that we're going to investigate. And so that's the state of the data that we have thus far. So I'd like to thank, and I know Dr. Thompson would like to thank everybody that's on the slide in great detail. But since for time's sake, so I don't have that time, I'd like to just thank everyone in the Thompson lab for all the hard work that they provide, the collaborators that we collaborate with, as well as the individuals in the IPSL consortia and LINCS consortia. I'd like to personally thank my advancement committee for all the help that they've provided me over the last few years. And special thanks to our funding sources, as well as the Genomic High Throughput Facility, which has been really helpful in quickly getting the data to us, as well as helping with some of the experimental design. Jenny Wu in particular has been very helpful with the bioinformatic analyses, and Melanie as well too. Thank you very much.
Questions for Ryan? So I'm not sure if you're aware, but actually RAS, which has another way in RSM, is a repressor of NeuroB1. So have you looked at whether it's actually detecting in your cells of the HD neuron information, whether you can detect an RSM finding in the body of NeuroB1?
We have not. That's something that we should probably look at though, but we haven't looked at that yet.
Patient samples that have different number of repeats and do the iPS eventually neurons. If you see more feedback or difference in higher repeat samples?
Somewhat in some of the functional assays that we see. What I didn't show in the slides is actually we did some immunofluorescence analysis and actually some serological counts to actually determine throughout the differentiation, are we seeing any large gross differences in the amount of populations of the neurons that we are obtaining? And we actually don't see that, specifically in the HD cells, we don't see that at all. So that's really interesting. But at the functional level, when we do like cell death assays, as well as at the transcriptomic level, we do see that there are some genes and some assays that correlate really well with the higher repeats. So it is
Are there sequence poly-Q repeats that are encoded not just by CAG, but by CAG and CAA? I believe that's the right one.
yeah, there's actually other diseases that have different types of potential expansions.
Okay, but I mean here in Huntington, I mean, do you observe any
Oh,
cases where it's not just CAG?
No, at least not to my knowledge. I believe that with Huntington's disease, that CAG repeats the only repeat for that front exon portion.
There's a mouse model that has one.
So if you intersperse some CAs, is it protected?
No, the mouse model is the back mouse, so it's behind the disease, but it's still a
Okay, I mean, I'm just thinking that the expansion would be aggregated, like a normal repeat, like with the CAs would prevent more expansion.
It doesn't matter whether it's mixed or single or triple repeat, but to do this and have instability, you navigate it, you start mixing it,
Right. So are there protected populations that have this as, okay. Well, I suppose if people haven't looked that much, right? All right, thanks.
Thanks, Ryan.